Hi, my name's Emma Funk. I'm a third-year PhD candidate in the lab of Dr. Dennis Guttridge here at MUSC, and I'll be going to AACR this year to present my work on GDF-15 and GFRAL in pancreatic cancer development and immune cell suppression. GDF-15 was identified by our lab a couple years ago as a cytokine that is very upregulated in pancreatic cancer patients and is known to be an immunosuppressor. Now, what's interesting about GDF-15 is its most well-known and agreed upon receptor is a receptor called GFRAL, which is almost exclusively expressed in the brain of individuals. Now, the role of GFRAL is primarily to modulate appetite through induction of nausea, which at a glance may not seem like it's very related to immunosuppression, but we actually see a really cool natural example of this in pregnancy. Now, women, when they become pregnant, have GDF-15 upregulated, and this is what is known to cause morning sickness in these individuals, but also at the same time, it was discovered that GDF-15 plays an important role in embryo implantation by actually suppressing the activation of these T cells, which is kind of cool because in pancreatic cancer, we see something similar. Now, pancreatic cancer patients tend to have a high rate of cachexia, which is a body weight loss that happens when patients have cancer. And what clinical trials have shown is that inhibition of GDF-15 is actually able to reduce nausea these people experience and help them regain their body weight. Now, what hasn't been explored is how targeting this signaling axis could increase T cell efficiency in pancreatic tumors. We wanted to ask the question of whether this GDF-15 GFRAL signaling axis was involved in pancreatic tumor development and immune cell suppression, especially because we discovered that T cells don't even express this GFRAL receptor. My work I'll be presenting shows that GDF-15 actually is able to directly suppress the activation of T cells independent of GFRAL. And then using mouse models where we have knocked out GFRAL, we see no difference in pancreatic tumor development or immune cell infiltration into these tumors, suggesting that GDF-15 may be modulating the tumor microenvironment of pancreatic cancer through a GFRAL independent mechanism, leaving exploration into what receptor this cytokine could truly be signaling towards.